Muito obrigado. Eu acho que, falando de cá, a gente não precisa de máscara, não, né? Eu sou pobre do palestrante, para falar daquele estudo é mais difícil. Mas muito obrigado a todo mundo. Nós estamos com outra auditória, assim, de gente também, para poder assistir a nossa apresentação de hoje. Hoje o seminário, nós vamos contar, let me speak in English. We will have today a talk, a seminar given by Professor Chris Pittenger, and sometimes also known as Chris Pittenger, and and he is a professor of psychiatry at University of Yale, and I will not spend much time presenting him because he's going to speak about what type of work he does there. And this event is organized by IEAT, this guy here, the Institute for Advanced Transdisciplinary Studies. And we invite you to learn more about the activities of IEAT, because we are always trying to bring some interesting people here. There's one activity that we'll have next month, I guess, that will be in June. Will be a professor from University of California, Santa Barbara, who is going to talk about Roosevelt's trip in the Amazon together with Coronel, como é que ele chama, gente? O Rondon. Coronel Rondon. Né? It will be quite interesting. And this professor is a graduate from our university here. And then let's then get Chris to speak here. I would like to thank you very much for coming here, for letting us learn a little bit about your activities there in Yale. Thank you. O Reni quer saber quem é da psicologia. Those are the psychologists. Tem alguém da engenharia, hein? Que fez engenharia no passado não vale, né? Tá certo. Ok, thank you.
Thank you. Thank you all. It's a pleasure to be here. I'm here in part because my sister is a professor in the music school. She is here. And she promised to tell me if I talk too fast, because I usually talk too fast. So you let me know, okay? And let me know if I need the microphone. Usually, no, but let me know. Okay. So I was asked to start by telling a little bit about myself, where I come from, so you know who you're listening to. So that's me 12 years ago. So I have always been interested in understanding the brain, understanding how it works, cells, molecules, networks, but also understanding what it does. Thoughts, consciousness, feelings. And so these have been my shared interests for a long time. I did my medical doctorate and my PhD at Columbia University in New York a while ago now.
Great.
Yeah.
If you could use the microphone, just because we are transmitting through YouTube, and so that, yeah.
No pressure. Okay. Hello again. So I've long been interested in how the brain works, but also what the brain does. And what the brain does isn't just move my feet around. What the brain does is also think and feel and reflect and ask questions about what the brain does. Right? So anyway, so I did my MD degree and also my PhD in neurobiology at Columbia. And I did my PhD with this guy, Eric Kandel, who is a psychiatrist who did neurobiology research, and he won the Nobel Prize while I was there, which was kind of fun. And he studied how the brain changes due to experience, right? So when we have experience, when you listen to me talk, if you remember anything, which I don't assume because it might be boring, but it actually changes things in the brain. things that with the right tools you can see, things that you can test. And that's how information is stored in the brain. I thought this was amazing because that's how the brain connects with the world, right? That's how we learn and change and develop in response to our experiences and the situations in which we grow. So I thought that was fascinating. So I stayed for a little while. I got interested in learning about habits. And habits are things that when we learn them, they're not instincts, they're not automatic. But once we've learned them, they become automatic. They become efficient, very quick. That's good. They become inflexible. And sometimes that's bad. So I got interested in that. Anyway, I went to Yale University to do the rest of my psychiatry training. So in the United States, it's 4 years of college. I did 4 years of medical school, 5 years of PhD, then 4 more years for psychiatry. So I was about 67 by the time I finished. No, I was about 35 by the time I finished. And I became through a long, boring story, I became the director of the Obsessive Compulsive Disorder program. And I think of obsessive compulsive disorder, which I'm not going to talk about very much today, but I think of it as cognitive habits, habits of thought that are too inflexible, that have gone bad. So that's how I got into this. Anyway, so that's where I landed. Whoops, other way. So I've done a lot with obsessive compulsive disorder. That's my book about obsessive compulsive disorder. Not going to talk about it today. But in the obsessive compulsive disorder group, we do all sorts of stuff. We look at new drug treatments. We look at new psychotherapies. We look at brain stimulation. We look at animal models. We look at lots of stuff. Each one of these is a different lecture some other day, except for the new drug treatments, which I'm going to talk about a little bit today. I also do a lot of training. I run a training program. I'm actually, after this, in a couple of days, I'm going to Sao Paulo to talk with the psychiatry department there. They're always sending MD psychiatrists to us to train. So I do a lot of that. But then recently I've started to do this, and this is what I'm going to be talking about today. And the reason that I look at psychedelics, the symbol of Yale is a bulldog. So I found the psychedelic bulldog. This is a bulldog who's been using a little bit too much LSD. I didn't draw it, but anyway, the reason I got interested in psychedelic research was because of this guy. His name's Ben Kelmendi, who I've been working with for, 16 or 17 years, since I was a resident just learning psychiatry, and he was in college. And we worked on OCD for years and years and years. He became interested in psychedelics, and he taught me about psychedelics and brought. So I always want to give him credit. And that emphasizes how we, the professors who are starting to get a little gray around the edges, learn from our students. Because I would not be in this work at all if one of my students hadn't come. He's now on the faculty as well. Now we're colleagues, but started as my student. Those relationships are really important, and I like to acknowledge. Okay, so that brings me back to, that's enough about me. Brings me back to my title, Psychedelic Treatment for Psychiatric Disorders. Is this a new frontier? Well, it's not. I'm going to define these things. I'm going to define what I mean by psychedelic. We'll get to that. But I want to start by saying the new frontier. There is a lot of excitement, at least in my country, and I assume here, there's a lot of people talking and researching and getting very excited about using drugs, especially psilocybin, but also MDMA and LSD and ayahuasca and other. I'm going to talk about these in some detail as we go through. A lot of excitement about them, and we call it a renaissance. We call it a new frontier. Whoops, I keep pushing the wrong button. It isn't really a new frontier because these chemicals have been around forever. What's shown here is psilocybin comes from the psilocybe mushrooms, which have been used in rites by various peoples, mostly in this country, but there are also psilocybin containing, in this hemisphere, but there are also psilocybin containing mushrooms in Europe and Asia. They haven't been used as heavily. They're most famous in this region, the Sierra Mazteca in Mexico, where taking psychedelic mushrooms as part of rituals has been an essential part of the religious practice and the culture for hundreds, if not thousands of years. I am not an expert on this. I am not an anthropologist. I do not know all this history, but I acknowledge it. Of course, the same is true in other places, including in this country. So ayahuasca, which I'm going to talk about just briefly later in the talk, is a tea that's brewed from actually a number of different, but at least 2 different plants, and used by various groups in the Amazon basin to produce psychedelic and mood-altering effects. Again, for hundreds of years. I can't tell you the history. I hope there are probably people on this campus who can, but for hundreds of years. So I am talking about the modern medical use and rediscovery of these chemicals, but they have been around for a very, very long time. In Western scientific experience, they've been around also for hundreds of years. The first published account of a European who came to the New World, and used, I actually don't know if it was psilocybin or peyote or what chemical it was, but wrote about his experiences so that other Western scientists and doctors, read about it. It goes all the way back to 1591. Psilocybin in 1799. Again, this is just the Europeans discovering them. They were there all along, but this is when the Europeans discovered and began to write about them in a Western medical context. And then in the 20th century, all sorts of things happened. Mescaline was found in cactus. That's the active ingredient in peyote, which is used, which comes from cactus grown in Texas, is used in some Native American rituals. MDMA and phencyclidine aren't found in nature. These are new chemicals that were made in chemistry labs, mostly in Germany and Switzerland. This guy, Arthur Hefter, was a giant in the early field. I'm not going to go through all of these, but you can see that through the beginning of the 20th century until 1960s, very active. Lots of new chemicals being either discovered from the natural world or synthesized. Lots of people taking them, discovering they did interesting things to their minds. And very famously, Albert Hofmann, who synthesized LSD in 1938, left it in a tube in the refrigerator. And then in 1943, he accidentally got a little bit. It's an incredibly potent substance. He got just a tiny bit, got into, I think, through a break in his skin. And he began to see colors and began to feel connected with nature. He drove a bicycle through Zurich in Switzerland. I don't know how he didn't run into anything, because it's a very famous bicycle ride. Anyway, so then in the 1960s, these substances were being investigated by the medical industry. Bayer had LSD, Sandoz had psilocybin. They were being marketed and used, even though the science was just beginning. But they were available. But at the same time, these substances, especially LSD, were being used for lots of other reasons. Recreational use, very famously, Timothy Leary at Harvard in the United States felt that everyone should be on LSD. It stopped being scientific and became an almost religious crusade. It got tied up with the hippie movement. That made lots of people very scared, lots of old people very scared, and they reacted against that. And so in 1970 in my country, and around the same time across the world, very strict laws were passed to restrict the use of these chemicals. And I'm not going to go into safety. I believe that we should have careful restrictions. I think that we should treat them as a medical problem, not a legal problem. But I do think we should have restrictions on heroin, on cocaine, on these things. Psilocybin and LSD were not dangerous. They're not addictive in the same way those are. Now, you shouldn't go riding a bicycle while you're on LSD. That can be dangerous, but they're not addictive in the same way. The reason that they were caught up in these legal restrictions in 1970, 1971, 1972 was because people were scared of them, scared of what they represented, scared of what they meant. And so it has taken us 50 years to get back to taking these molecules seriously as something that can help people because of that fear. All right. What is a psychedelic? I promised I would say, what is a psychedelic? The answer is, you'll get different answers depending on who you ask. Some people will say a psychedelic is these chemicals over here. Psilocybin, 5-methoxy-dimethyltryptamine, ibogaine. Those are the psychedelics. Some people say that. Some people say a psychedelic is anything that if you take it, it does weird things to your consciousness, right? And everything on this slide is an example of that. Okay, I use the word classical psychedelic for these. Also, I include LSD. This is synthetic, doesn't occur in nature. I use the word classical psychedelic for these. We think they act in a similar way in the brain. I'm going to talk about this. It's binding to a particular serotonin receptor and causing a particular set of changes in the activity of the brain. And they, in terms of the subjective experience when one takes the drugs over here, they're fairly similar. That some of them are quicker, some of them are slower, some of them are more intense, some of them less. But they're fairly similar. So I use the word classical psychedelics for these and atypical psychedelics for these. Some people use the word empathogen, which means a chemical that makes you feel connected to the world, for MDMA and mescaline. And these are, like, ketamine is a dissociative anesthetic. It's used, so different words are used. But I use the word psychedelic for these and atypical psychedelic for these over here. I'm going to talk about both. Most of what we're going to talk about is about psilocybin today, and that's because that's where most of the research over the last 20 years within the Western medical establishment has been. There's also been a lot on MDMA. I haven't done any of that myself, and I will talk about that a little bit less, and I'll just touch on some of the others. I should have said at the beginning, I'm perfectly happy to have people interrupt me. If there are questions, feel free to raise your hand, throw things at me, yell out. It's all good. So if anything I say doesn't make sense, or if you have a follow-up question, don't hesitate. And I believe we will have time for questions and discussion at the end as well. In fact, I've ended my talk with some questions, with big open questions, not a conclusion. So we'll have time to talk about those. Okay. So this is a list of some of the events of the psychedelic revolution or renaissance in the modern era. It goes back to the work in the 1990s of some true believers. So there were a few people who never stopped being excited about and researching the psychedelics, especially Franz Vollenweider in Switzerland, who just retired, was working throughout this time. The restrictions in Switzerland weren't as harsh as they were in most other places. So a few people were working all along. But in the 1990s, some scientists and some philanthropists with money began to, saying, how are we believe that these drugs can help people. Some of them said, we believe that these drugs can fundamentally change society and create a revolution in consciousness. That's different, but medical people in the medical field were saying, we believe these drugs can help people. How can we get them back into the discussion, into the conversation, into the research? They formed a group called the Hefter Society that began to, after that guy I mentioned in the other slide, Hefter from the late 19th century, that began to fund this kind of work. And a pioneer, Roland Griffiths, who's really a brilliant man, at Johns Hopkins University in the United States, started his program at Hopkins studying psychedelics in 1999. This was a very scary thing to do, right? They were illegal. No one thought that it was real science. He's really remarkable. So I've come into this field very late, when everyone's excited about it. That doesn't require any bravery at all. Roland Griffiths had a lot of bravery to come in here in 1999. And then Robert Carhart Harris is another early pioneer working with David Nutt at Imperial College in London. Same thing, came in when no one else wanted to take the work seriously and began to push it. Roland Griffiths is actually ill and is retiring now. Robert Carhart Harris continues to be very active. So the first modern study of psilocybin published in a journal that scientists took seriously was in 2011. I'm sorry, first study in psilocybin as a treatment. Okay, there were studies in the space between here in psilocybin in healthy people to characterize its safety and characterize what it does psychologically. But the first study of psilocybin in a treatment was in 2011. MDMA, which is also called ecstasy, was first studied, published in 2011. I'll be talking more about both of these lines of work. And then in 2016, there was this enormous year where some studies, which I will show you, in depression and also in the despair and anxiety of people late in life with cancer as a treatment for their psychological distress. These were published in good psychiatric journals in 2016. Suddenly everyone was talking about it. And from then till now, it's been an explosion of scientific interest and popular interest in the field. And that's shown in this slide. So this is studies that have been registered, not yet published necessarily, but registered as official government-approved studies that are in process for MDMA. This early work was all for MDMA, also called ecstasy, in the treatment of PTSD. And I'm going to talk about that a little. That goes back 10 years. Then the psilocybin studies started. And then, boom, in the last 5 years, there's been a huge number of new studies. I think 2 of those dots are me. So this is a very big, broad-ranging thing. And it's continuing to grow. Okay. So the next thing I'm going to do, and it's going to take me half an hour, is I'm going to go through the different psychedelics that have been studied in recent years, most of our time on psilocybin, but a little bit on the others, and just bring you up to speed with how these have been used. And my focus here is really very much on treatment. Okay, as I said at the beginning, is this a new frontier in the treatment of psychiatric disease? I'm a psychiatrist. I like to help people. The goal is, do these things help with treatment? It's a whole nother lecture to talk about what do they do to the mind, what do they do to the consciousness, what do they do to religious feeling, to sense of connection with others, to sense of connection with the natural world. I'm less expert in those topics, and it's not what I'm talking about today, but we can certainly discuss at the end. I'm going to be focusing on treatment. So I'm going to start with LSD because it's the most famous, at least historically, the most famous. So as I told you, LSD was developed by this guy, Albert Hofmann, a Swiss chemist working for Sandoz, great big Swiss pharmaceutical company. He made thousands of chemicals. There wasn't anything special about LSD until that bicycle ride in 1943, when it jumped off, it became clear that this was something really special. This is the molecular structure of LSD over here. So discovered in 1938, hallucinogenic properties discovered in 1943, lots of research through the 40s and the 60s. And this is important. It wasn't just used by hippies looking for a good time in the 40s through the 60s. There was quite a lot of careful research done by psychiatrists and neurologists in this early era. By modern standards, by my standards today, they weren't great studies. But this was 50 years ago, right? By the standards of the day, this was good science. And there was good evidence in the treatment of pain, in the treatment of alcohol use disorder, in the treatment of depression. There was, pretty good evidence. But then, as I said before, the recreational use led to a backlash. Am I supposed to be in the picture, or am I supposed to be? So what does LSD do? And everything I'm going to tell you here also applies to psilocybin, so I'm going to spend just a little time talking about what LSD does. LSD is what we call a dirty drug. That means, so a clean drug would go in and do one thing. It would attach to one target in the brain. It would have one effect on neurons. That would be a clean drug. LSD is a dirty drug. It sticks to lots of different things in the brain, which makes it a little harder to figure out how it does what it does. But a lot of research, actually some very early research done by a senior colleague of mine who's still there, George Agajanian, in the 1960s, found that LSD binds to a particular receptor in the brain. So for those of you who aren't biologists, our cells are bounded by this fatty lipid membrane that has receptors in it, okay? The membrane by itself doesn't do much. It keeps stuff in, it keeps stuff out, that's about it. All of the action is in the receptors, because the receptors can let stuff in, they can let stuff out, they can cause electrical phenomena, they can cause signaling. This is how neurons work, right? Things bind to receptors, they cause a change in the dynamics of the membrane, changes in the flux of ions across the membrane, that causes electrical events, that sends signals through the brain. That's how neurons send signals through the brain. So most of our drugs in psychiatry bind to receptors. Not all, but most. So it turns out what George Agajanian found is that LSD binds to the serotonin 2A receptor. So this is a cartoon. The receptor goes zipping in and out of the membrane like this. This is what the actual thing looks like. This was just solved a few years ago, that they found every one of these little balls is an atom. So this is the actual structure. You'd have to imagine that the membrane is going back and forth like this. And LSD, actually, I cheated. This is a psilocybin here. But LSD binds to the same place as psilocybin, right here in the middle, and it activates the receptor. So it activates the serotonin 2A receptor. And that does a bunch of stuff to the cell that I'm not going to talk about today. But this is what we think happens. This here, I'm going to move this over a little. So this here is a picture using positron emission tomography of where the serotonin 2A receptor is in the brain. Okay? So some parts, this is the outside of the brain. This is a horizontal section of the brain, like this. They didn't actually cut the person's brain to get this picture, just to be clear. Okay, this is put together in a computer. But this is a horizontal section. This is a coronal section, and this is a sagittal section like this. And if we had 20 weeks, we could do a neuroanatomy course, and I could take you through all the different parts of the brain. I'm not going to do that today. But you can see that there's more binding in some places and less in others. There's a lot of binding here. I want you to remember this. This is the sagittal section. And there's a lot of binding here in the middle, in the front. And there's a lot of binding here in the middle, in the back. Okay, I'm going to come back to that, so remember that. So that's where the serotonin 2A receptor is. And when LSD binds to the serotonin 2A receptor, it fundamentally changes the firing patterns of neurons in the brain for a period of, in LSD, 8 hours. Okay, it's a long time. What this is a picture of, this is a functional MRI picture. This was done by Franz Vollenweider in Switzerland, together with Alan Antisevich, a colleague of mine at Yale, did this analysis. And Katrin Preller, who did most of the, she did most of the actual analysis. The others just paid the bills. But what they did is they measured people's brain activity when they're just lying there in a brain scanner. Then they gave them LSD, and then they measured it again while they were having interesting experiences. And then they compared the two in every person, okay? Now, what's shown here isn't brain activity. It's a measure of how different parts of the brain are talking to each other. I'm not going to go into the technical details because that would take more minutes than I want to devote to it. But it's a measure of brain organization. Blue means less connected. Red, yellow, orange means more connected. And what you can see is that these regions that I asked you to remember are very much less connected on the LSD. Whereas this is visual cortex back here, very much more connected. Well, they're hallucinating, so that's not that's kind of makes sense. But this is very interesting, that it's doing something, and also some regions over here which actually are functionally related, even though they're in a different place, they're part of the same network, they're functionally related. So it's doing something to these regions. Well, that's very interesting because those regions are part of a network of brain structures that's been studied now for 20 years in functional neuroimaging and cognitive psychology studies called the default mode network. And I'm going to say just a few things about the default mode network. That, again, is a whole nother lecture, but it's quite interesting because the default mode network is what turns on when you're not doing anything, okay? So if you have someone lie in the scanner, their default mode network's kind of active. You then give them something to do, something to look at, something to respond to, something to say, it goes down, and other parts of the brain come up. And that's led to the idea that the default mode network is the part of the brain that we use to reflect inwardly, to daydream, to think about ourselves, perhaps to form our own sense of our being, who we are, okay? And the LSD is disrupting that. Not permanently, but while the drug's on board. And that makes a certain amount of sense with some aspects of the LSD experience, which involves a feeling of connection with the world, breaking down of the boundaries between oneself and the world. Sometimes people use the word oceanic boundlessness. I don't know what that means, but just this sense of connection with the rest. Well, if you're breaking down the structures and mechanisms, not the structures, but the mechanisms in the brain that are responsible for helping you define who you are in relation to the rest of the world, well, that kind of makes sense. Is it true? I don't know. But it kind of makes sense and is the basis of some of our current hypotheses about how these drugs work at the network level and at the psychological level. So modern research on LSD is fairly limited for three reasons. One is people are scared of it. Okay, this is the drug that triggered the panic in 1970. So psilocybin is a little safer. 2 is it's kind of a dirty drug. 3 is it lasts too long. Okay, so for these reasons, there hasn't been that much modern research on LSD in treatment of psychiatric disorders. There is some evidence that people who use LSD occasionally are happier, that it's good for mood. If you go to a festival, the big one that has been studied in the United States is the Burning Man Festival, where people are using all sorts of drugs, and you do lots of surveys. The people who are using cocaine or marijuana, they're kind of stressed out and not necessarily having that great a time. The people using LSD and psilocybin are systematically having a better time. They're happy. So there's some evidence that this can help with mood when used casually. Now that's always a little tough because you don't know what people were actually using, what the dose was, how pure. It's not a great way to do science, but it's interesting in terms of recreational use. Some people are looking at microdosing, taking teeny amounts. There's very few studies of that in terms of treatment, and that they aren't very promising. So there's not that much with LSD. There's a few studies coming on. We do need to be cautious with LSD. I don't want people to leave this lecture today thinking that I think everyone should be taking LSD and psilocybin, that I think we should put it in the water supply. I think if I did say that, they'd kick me out. They'd cut off right now. So that's not my message. The studies that I'm going to share data with you on in the treatment of depression and alcoholism and obsessive compulsive disorder are done in very carefully controlled settings with very pure drug, with medical monitoring. In those settings, it's safe and it seems to help a lot of people, and I'm going to show you data for that. That's a totally different question from eating a handful of mushrooms at a rock concert, right? So my view is that we should go very slowly. I don't have any problem with people using these things recreationally, but I don't think it's benign. And I certainly don't think you should do it if you're sitting on a rooftop. So that's not what I'm espousing. What I'm espousing is taking these things seriously as potential treatments, as things that can help people. That's what I espouse. Uncontrolled use in recreational settings. If you do surveys, about 30% of people who've used LSD say they've had a bad experience. And sometimes that can last days or weeks. And sometimes, rarely, very rarely, that can last a very long time, okay? It's less than 50%, but it's a lot more than 1%. It's 30% of people have some negative experience. Again, that's out not knowing how much they're getting, how pure it is, and the environment, that those things aren't controlled. It's very different when we do it in the lab. More research is needed to understand this. But as I said, there's not that much work going on in LSD compared to psilocybin. Psilocybin is where the bulk of the research has been over the last 20 years, and I'm going to spend a little time showing you some data. So psilocybin, as I've already introduced to you, comes from these psilocybe mushrooms. There's like 200 species of mushrooms that contain psilocybin. This is the molecular structure here. It looks an awful lot like serotonin, the normal molecule that binds to the 5-HT2A, the serotonin 2A receptor, but we're not going to go into the chemistry today. Comes from here. I already told you this. Introduced to North America in the 1950s. I basically already told you all of this. There were some early studies. They look pretty promising. There was more work in the 50s and 60s on LSD, a little bit less on psilocybin. But then this is where it really picked up in the 1990s and the beginning of this century. So here are some pictures of the brain of people using psilocybin, and they may look kind of familiar because it's awfully similar to LSD. Okay, what's shown here, this is where, this is PET imaging, positron emission tomography imaging. That allows us to visualize specific receptors in the brain of a living human being using a radioactive tracer. So if I put a radioactive tracer into somebody that binds specifically to the serotonin 2A receptor, this is what it looks like. It's a different, on the left. It's a different study than what I showed you before, but it's showing you basically the same thing as what I showed you before. If I then give that person psilocybin, this is what it looks like. Why? It's because the psilocybin is sticking to those receptors. It's kicking the radioactive tracer off. I can't see it anymore. So this confirms that the psilocybin binds to that receptor in people, in living people. Another thing, the data isn't here, but if I give psilocybin to people at an appropriate dose, they have an interesting experience. If I then give them a drug that blocks the 2A receptor, it goes away, does nothing. So that gives me evidence that this shows me it binds to the 2A receptor. What I just told you shows me that that binding matters. It's important for the psychological effects of the molecule. Okay, so these studies have all been done. What's shown here is from a study of people with depression that I'm going to tell you more about in a second. This is what happens when I treat someone with depression with psilocybin. Where is the brain perfusion? Where is the brain activity changed? And what you can see, it's a little messy if you're not used to looking at these pictures, but here along the middle of the sagittal section, you can see a reduction in the blood flow going to the default mode network. You can also see some changes here in the lateral cortex that's also actually connected to the default mode network, and then in some other regions that aren't. So it's not super specific to the default mode network, but it does affect it. So qualitatively similar to what I showed you before. This is the same thing, but done with that brain connectivity measure in an fMRI magnet, okay, by my colleague Alan Antisevich and Katrin Preller in his lab. And it looks very, they did the same experiment with LSD and psilocybin. It looks very similar. They have a reduced, not activity, but connectedness in the default mode network, shown here, increased in the occipital lobe, which is dedicated to vision, and then some other changes more broadly in the frontal lobe. This is just to remind you that the default mode network is a way that we think about these and try to organize these, even though it remains a hypothesis, like the idea that these drugs, because of the distribution of the serotonin 2A receptor, have a disproportionate effect on this network, and that may explain some of their psychological effects. This is a hypothesis, but it's one that I think is quite interesting. Now, we also have some studies of what psilocybin does in the brain in the long term, right? So if I'm interested in how does psilocybin change people's sense of religiousness or connection or spirituality or hallucinate, then I want to know what's happening while the drug is in their body, right? But if I want to use these drugs as a treatment, where I give someone the drug, and then I want them to be better a week or a month or a year later, okay? I don't care what it's doing. And I mean, of course I care. I'm a scientist. I want to know things. But as a doctor, as a clinician, I don't care what it's doing while the drug's on board. I care what it's doing a week, a month, a year later. Right? And we're starting to have some evidence about that. This is a study from my colleague Alex Kwan at Yale, studies in mice. So this may be a little hard to see. This is a mouse that's in a little tube, its head is fixed. They do this slowly. The mouse gets pretty used to it. So by the time they're doing the experiment, it doesn't seem to mind too much. And they use this microscope, very fancy microscope, to look into the brain in the mouse while it's, sitting there running around on a ball. And they can look at specific neurons. This is a slice through the mouse's brain, looking at specific neurons that have been made fluorescent, just so you can see them. Okay, and they can visualize those neurons. This is a branch from a neuron. You zoom in to this here, and it looks like this. These branches of the neurons have these little bloops, these little synaptic connections. They're called dendritic spines. They are the sites of synapses. They're the sites where neurons are talking to each other. And when you give psilocybin to a mouse, so this is the beginning, this is baseline, nothing's happening. This is 1 day after, 3 days, 5 days, 7, 31 days, or 34 days, I guess, after psilocybin. They count the spines. You get more of them. You create more synaptic connections in the brain. After psilocybin, starts at day 1, lasts for a whole month. Okay? That's kind of cool. Does this cause antidepressant effects, whatever that means in a mouse? I don't know. But it's interesting that the drug, one dose of the drug, is having long-lasting effects. And it's also interesting that similar effects on dendritic spines in the same place in the mouse's cortex are seen in response to ketamine when it's used as an antidepressant and to certain other chemical antidepressants that we use. So there's some consistency there that suggests that this mechanism might have something to do with the ability of these drugs to stabilize and improve mood. So that's interesting, and this is very active work now. The field's moving very quickly to try to understand. We know what the drugs do at the level of the receptor. We know a fair bit about what they do at the level of the cell. What do they do at the level of the synapses, the network, the whole brain? That's where there's a lot of work going on now. All right. So now I'm going to shift to clinical studies. And I'm going to tell you a bunch of studies, 1 slide per study, most of which are not mine. Okay, this is mostly published work, and I've got the citations on all of the slides. But first, I want to tell you how we do these studies. Okay, it's complicated, and I told you I was going to end my talk with some questions for discussion. One of those questions is, do we really have to do it this way? Because what we do is we have someone come in, they lie in a comfortable bed in a peaceful environment. This is actually my office, because we were going to be doing it in a regular dosing room. We had a COVID outbreak. We had to shut down that room, so we went and did it in my office. My office doesn't usually look like this, but we get people comfortable, relaxed. We have them sit with 2 attendants who they have gotten to know, they've spent time with before. They're told what to expect. We have nice soft music playing. We give them eye shades. We do check their blood pressure and stuff like that too, but we make it as unmedical as possible. And these 2 attendants sit with them throughout their psilocybin experience, which lasts between 4.5 and 7 hours. Okay? That's fine. It's a fine way to do research. People generally have a positive experience. It keeps them safe, both psychologically and physiologically. All good. There's no way we can actually go out and help a lot of people if this is what it takes, right? It takes too much space. It takes too much time. It takes too much person hours. All right? So that's a problem. But anyway, this is how we do the research, and this is how my group does it too. Everyone's done it this way for 20 years. Some people are deep in the field, are deeply invested, and this is important. This is important for shaping the psychological experience. Otherwise, it'll never work. Other people think that that's all ridiculous, and you should just sit someone in a chair in a corner and let them do their thing. Obviously, if that were true, it'd be much more efficient. I'm somewhere in between, so this is one important open question. But this is how we do the studies. We lie people down. They're sitting with 2 people. It's always 1 male and 1 female, unless they explicitly ask for something different. They're both always there, and they don't do much. Like, people on psilocybin are they're mostly inside their heads. So the 2 people don't do that much. But if the person becomes frightened or becomes disoriented, then they're there to help. Okay. So the first studies that were published in the Western medical literature in the modern era, and the first I'm going to tell you about, were done in patients with advanced cancer. Okay, patients who probably didn't have that long to live. And obviously, doctors tend to be very focused on medical health in those contexts, but the psychological experience of contemplating your own death, perhaps together with, low energy or nausea from treatment or pain, are profound. And if you can't extend someone's life, but you can reduce their psychological suffering, you've done a really good thing. So that's what the focus of these first studies are. There was some data on this from back in the 50s and 60s, and that's one reason that the early focus, this is mostly by people at Hopkins, UCLA, and NYU, not in my group. We weren't on the field yet. But the one reason that they focused on this is because there was data and it's a huge clinical need, and it was a particular passion for some of the clinicians who were doing the work. But another reason is it was a lot easier to get the regulators to agree to give psilocybin to people who were dying and were suffering than it was to get them to give psilocybin to someone in their 20s who was depressed, right? So this is the sociology, the politics played into this. This is where the first work was done. The first study was in 2011, and I don't have the data for that. It was a very small sort of proof of concept study. But then in 2016, these two hugely important studies came out, one from the group based at Hopkins and one from the group based at New York University. And what they did, so what you see in these graphs is on the y-axis, this is a scale of how anxious people are, and this is a scale of how depressed people are. Okay, we can't do a brain scan or a blood test to measure anxiety and depression. We just ask people questions, but this is carefully designed, well-established, proven scales to take people's experience, have an experienced rater ask them questions, rate their experience, put a number on it. So that's what the y-axis is. The x-axis is time, and the green letters, which you probably can't read, but this is where they gave the drug. So this is anxiety scales and depression scales before they gave the drug and after they gave the drug, going out 1, 2, 6, 7 weeks. And what you can see is that the people in purple who received the psilocybin together with some general support, not psychotherapy, but general support, got substantially less anxious and substantially less depressed. And the people who had exactly the same experience, well, no, they didn't have the same experience. You can tell. You know, we give people a placebo, it's kind of a joke because everyone knows if you're on psilocybin or if you're on, a sugar pill. But anyway, but they did have the same number of hours sitting with the same people, the same follow-up. They did not have maybe a little bit of benefit just from the close attention, but no lasting benefit. These are dramatic. I mean, this is a 50, 60% reduction in people who are suffering. This is a dramatic effect. And the fact that after a single session, it lasts for 7 weeks. And in fact, if you call the people back a year or two later, some of them have died. Those who have not continue to feel that this is really an important event. So this really took the world by storm. This is super important. This is another study, slightly larger study, about 50 people, but a very similar one. They gave 2 doses of psilocybin, a low dose and then a higher dose. And this is how many people, these are both depression scales. There's lots of data in these papers. I just picked out a couple to make the point. This is, they looked at what percentage of their people were not depressed anymore. They had been depressed, they weren't anymore. 5 weeks after or 6 months after a single session with psilocybin. And what you can see is that after the low dose, they got about 30% of people better. Some of that may be real, some of that may be, it's real, the people are feeling better, but some of it might be placebo, might not be because of the psilocybin. But after the high dose, they got 95% of people no longer depressed at 5 weeks, okay? Let's see, this is response, so they were less depressed. This is remission. They aren't depressed. Their depression signals are the same as someone who is not dying of cancer, right? This is remarkable, and it lasts 6 months. So as I said, 2016, this is when everyone sat up and said, wow, there's really something going on here. The studies are ongoing, and that hopefully that'll become available as a general treatment, which is a whole nother complicated issue. So given that the drugs are illegal in most countries, what is required to get them to be available to people outside of either buying it from a guy on the street corner or being in a research study, which are kind of your option, or going to Jamaica or Amsterdam. Those are the options right now. So there's a whole bunch of legal processes that have to happen. People are working on them. 3 years, if the data continue to look good. All right, studies in depression. So major depression is a big deal. 17% of people in the world will get a case of major depression during their life, twice as common in women as in men. This number's probably gone up, 17% number, because of the pandemic. And it's a huge problem. I know it's a huge problem on university campuses in my country. I assume that's global. And we have treatments, and they help pretty well 50% of the time, right? So new treatments, this is a huge, huge problem. And there have been a number of studies in depression. And in fact, I said I'm hopeful that psilocybin will become available. At least I know most about it in my country. I don't know how the regulatory regimens work in this country. But I'm hopeful that it'll become available in 3 years in my country. It's going to be for depression. That's what's leading the way, because that's where the biggest need is. This is the very first study done by Robert Carhart Harris's group at Imperial College in London. This was not a controlled study. They took a bunch of depressed people. They gave them psilocybin. You unfollowed and see what happens. This y-axis is a depression scale. It's a different one than the one I showed you before, but it's up is bad and down is good. They gave people a dose of psilocybin and looked 1 week later. Each of the colored lines here is one person. Okay, this is not averages. The other graphs I showed you were averages. This is one person. And you can see the large majority of people had a dramatic drop, some down to 0 or close to 0, 1 week after psilocybin. A couple people didn't. You know, it's not 100%, but it's a lot. And then this is 3 months later. And you can see that most people who had a good response continued to have a good response. Looks similar to the late life cancer study, but in a clinical target that is, I mean, alleviating the suffering of people with late life cancer is hugely important. This is much more important in terms of the total amount of global suffering that could be alleviated. So this was an uncontrolled study in 2016. More recently, 2 studies came out last year in 2021, one from the Hopkins group and one from Carhart Harris's group, that were controlled studies. Okay, this is a placebo-controlled study from the Hopkins group, Roland Griffiths and his colleagues. Pivotal study, really important study. Again, the y-axis is how depressed people are. This is at baseline. This is pretty depressed, you know, not can't get out of bed, you know, but suffering. And then 5 weeks after the giving us a dose of psilocybin in the bed with the music, with the eye shades, with the people, and giving some support afterwards, they got this dramatic drop. This is barely depressed. You know, maybe not your best day, but barely depressed at this score. And that's stable out to 8 weeks. This is the people who got psilocybin later, at like week 10. So everyone got psilocybin in this study, but they either got it immediately or later. So again, evidence of a dramatic improvement. And this is a study, this is an interesting study. This is from Carhart Harris's group. Here they treated people either with psilocybin once or with Lexapro, escitalopram. I don't know if it's called Lexapro in this country, but it's a standard antidepressant. It's one of my favorite antidepressants if I'm treating people. It works pretty well 40% of the time. Side effects are usually mild, so it's a good, it's a good first thing if you're going to treat someone with a drug. The blue line is the improvement in depression in people treated with escitalopram. Okay, average improved. Some people improved a lot. Some people didn't improve at all. The average improved. Okay, that's what we expect. The orange line is people who got a single dose of psilocybin. Okay, these were not, when you do statistics, these 2 lines aren't different statistically, but the psilocybin's sure not any worse than the Lexapro, right? And it might be better. So that's pretty dramatic. And again, it lasts. Remember, the people with Lexapro are continuing to get the Lexapro every day. People with psilocybin got it once. So pretty exciting. All right, that's all I have to say about depression. The next is studies on alcoholism. There's a lot of interest in the use of psilocybin in the treatment of addiction in general. There's not nearly as much data as there is. The ones I started with the big ones. I started with the ones we had the most data for. There's not nearly as much data for alcoholism. But there is some, and in fact, there's quite a bit from the 50s. There was some really good work in the 50s and hundreds of people. Again, not rigorous by modern standards, but pretty good. So there's some ongoing studies there. The one that's published is from this group at NYU, led by my colleague Bogenschutz, and they looked at alcoholics and treated them with 2 doses of psilocybin. Now, in this case, I've told you that in the depression and the end of life studies, the psilocybin was given once with support in a comfy room, and then there was some support sessions afterwards, but it wasn't therapy. Right? It was just say, how you doing? Do you want to talk about how your experience? What does this mean to you? Sort of non-directive support like that. In this study, they did therapy. They did, careful therapy that is known to help some of the time with alcoholism, and then they did psilocybin in the middle. Okay? And what you see is people who were treated with psilocybin. So this is the beginning. The y-axis here is the percentage of days on which they had a drink, and the percentage of days on which they had a lot of drinks. And I don't remember what that quantum, if that was 5 or 10 or what it was. The red is a lot of drinks. The blue is anything to drink. So you can see that they were drinking 30 to 40% of days. Okay? Then, even though they were already in therapy. Then after psilocybin, that went out dramatically and went down to 10%. Now, these are averages. Some of these people stopped. They weren't drinking at all. Others were hadn't changed too much. The average went down dramatically. No control in this study. Now, I can tell you that Bogenschutz and his colleagues have followed up on this with a controlled study, a big one, 100 people. And I've seen the data, and they look pretty good. So there's better data coming. It's not published yet, and it's not mine to share. But this is exciting. So it's not nearly as advanced as it was as for depression, especially after those 2 studies that came out last year in depression, which are really good. But it's coming. And from the data I've seen, I'm optimistic. It raises an interesting question that I'm going to come back to at the end. That's kind of weird, right? That the same treatment delivered in basically the same way would be good for so many things. Now, it's not that weird, because as a psychiatrist, I know that doing unstructured, just generic therapy can be helpful, for a lot of different things. Then there's structured therapy that's very specific for specific, but unstructured therapy can be helpful for a lot of things. Lexapro can be helpful for a lot of things. We use it for depression, but we also use it, I'm talking too fast, slow down. We use it for depression, but we also use, you weren't yelling at me yet, but okay. But we also use it for panic disorder, generalized anxiety, PTSD, and so it's common in psychiatry for the single treatment to be non-specific in who it helps. So this isn't that weird, but it's a little weird, and that's worth keeping in mind when we think about how do these help. I'm going to talk about that a little more when I talk about OCD. Okay, so that's all the data I have to show on alcohol, but as I said, there's more out there. It's not publicly available yet. There's a bunch of other studies going on. These are in process. A lot started after that banner year of 2016. That's when more people got on the bandwagon. A lot of studies started. It takes a while to get legal permission to do the study. So it's a lot of work ongoing. Opiate use disorder, cocaine, depression in the elderly, anorexia, obsessive compulsive disorder. I'm going to spend, because I told you this is what I do. So I'm going to spend a few slides. We're doing the first placebo-controlled study of psilocybin in obsessive compulsive disorder, and I want to spend a couple times, because that's my privilege. I get to do that, so I'm going to. So I'm not going to spend a lot of time talking about what OCD is. I'm certainly happy to do that, but just trust me. It affects about 1 person in 40. It causes people to engage in rigid, ritualized behaviors, which they know are excessive and they know are doing more harm than good, but they can't stop anyway. And that knowledge actually is part of the suffering. Like, now, why am I doing this? And then I can't stop. Which, of course, is an experience we've all had with one bad habit or another, but in OCD, it's pervasive.
Toque, né? Toque.
So there was a single patient, there's now 5 patients in the literature who've been described who said, hey, I tried psilocybin and my OCD got better. So there was a hint, but just from patients telling us what their experience was. There was this one study in 2006 where they gave a bunch of patients with OCD psilocybin and they got better, but there was no control. It was only 9 patients. It was, so there's a little bit of literature out there, but nothing good. So we started to do the first trial. This is our trial. So this is just what the patients are doing. We screen them for a month. We get to know them pretty well. We actually admit them to the hospital. That's not because it's dangerous. It's because we want to talk to them every day and have a lot of time with them. We give them psilocybin on Wednesday, either psilocybin or a placebo. And then we follow them out for 12 weeks. Okay. And then the people who got the placebo, we give psilocybin later if they want it. So everyone gets a chance to try it, because I don't feel good about having people come to me for help and just giving them a placebo. That doesn't feel good. So everyone does, but it's either at the beginning or later. Okay. I thought I had another slide before I gave away the exciting part. Anyway, so then we measure their symptoms. Again, that's done with a questionnaire. It's called the Yale Brown Obsessive Compulsive Scale. It was developed at Yale in my clinic when I was 12, so I don't get to take any credit for it. But this is our first 9 patients. We're up to about 15, but I'm only showing the data for the first 9. Y-axis is the Yale Brown Obsessive Compulsive Scale. How bad is their OCD? Each line is a patient. The dotted lines are folks who got niacin, got placebo. Okay, it's not sugar pill. It actually does something, but very different from psilocybin. And the solid lines are people who got psilocybin. And you can see that 3 of these 5 people had dramatic responses. Okay, OCD doesn't respond quickly. I've never seen this before with any other treatment. It's only 3 of 5, so we'll see how the numbers work out. This is the most exciting thing. So this was 48 hours. The reason we did that is we wanted to know what happened before they started talking to their therapist, talking to their family, getting lots of interaction while they're still in the hospital. Okay, dramatic improvement. This is 12 weeks. So they only had psilocybin once. For the rest of this 12 weeks, we're not doing anything. We're just asking them, bringing them in, giving them the opportunity to talk, asking them how they are. I've never seen this with another treatment. Only 3 patients, but it's got us pretty excited. Folks with niacin, we only followed for a week. They knew they were on placebo, so then we then give them the opportunity to schedule a session with the real thing. And this is 3 of those 4 people who got niacin. You can see one of them didn't respond. Oh, I acknowledge 2 people didn't respond. I know these people very well. I have some theories about why they didn't respond, but it's not everyone. When we bring the niacin people back, this person didn't respond. This person responded briefly, but it didn't last. This person had a dramatic response. You put it all together, it looks like 50%. Okay? I'll take it. I'd rather have 99%, but it's pretty good. So this is ongoing. We'll publish this in 2 years, but we're pretty excited about it. This is we spend a lot of time with these patients. We record interviews with them. We have experts in qualitative analysis and linguistic analysis go through and score the interviews and look for themes so that we can answer questions that we hadn't thought to ask at the beginning. And these are some quotes. My life philosophy has evolved. I'm a lot less judgmental about myself and others. The impulse to engage in OCD symptoms feels unnatural now. I've still got those thoughts, but I don't know why they ever mattered. There is more to me than the defined me. There is an undefined self that I have found that wants to stay alive, to breathe, to laugh, to touch and feel, to awe. This guy wasn't a poet. He was kind of he really was kind of a taciturn guy who didn't talk about his inner life much. So this was striking. I put these up here, first of all, because they're kind of cool. They're pretty remarkable, actually, to listen to and to read through these transcripts. But in addition, I think this might get to the question of why does it help across so many things. So when people with OCD respond to a medication like Lexapro, escitalopram, or respond to psychotherapy, there's specific psychotherapies that work in OCD. Generic, how are you doing today, what's stressing you out, that doesn't help very much with OCD, but there's specific therapies that do. And people who get better after meds or therapy have a similar experience. They say, yes, I still have those thoughts. Yes, they still bother me, but they're not as bad, so I can live my life. Okay, that's what people usually say. People with OCD, after psilocybin say, oh yeah, I still have those thoughts. Yeah, they still come all the time. I just don't care. So it's changed not the symptom, but the relationship of the self to the symptom in a way that has allowed people to free themselves from the inflexibility that they were trapped in before. If that's what it's doing, suddenly, it's not so surprising that this could work across different psychiatric diagnoses. I mean, another reason is because psychiatric diagnoses aren't real things, right? They're sort of approximate descriptions of clusters of symptoms that go together. They're not diseases. That's another reason. But anyway, so I think this kind of, asking what a remarkable thing for a doctor to do, asking the patients what their experience is and paying close attention, I think can give us some insight into what's going on. All right, that's it for psilocybin. So that was the core of that was 30 minutes or more. That was the core of my talk, because that's where most of the action is. I'm going to more briefly talk about a few other things. I did mention ayahuasca at the beginning. So ayahuasca is a tea that is brewed from plants, originally in the northwestern part of the Amazon basin. Again, I don't know the anthropology very well. You need to find another lecturer for that.
Just to let them know. They know it more like as Santo Daime.
Oh, thank you. So ayahuasca consists of 2 different chemicals. One is called dimethyltryptamine. That was on that big chart that I showed you before, okay? We think it's quite similar to psilocybin. It acts quicker, it goes away quicker, but it has similar effects. We've got some folks at Yale doing studies on this, though I haven't done any myself. But the problem with dimethyltryptamine is if you take a pill, your stomach grinds it up. It doesn't actually get into your body. And it's not the acid in the stomach, it's a specific enzyme called monoamine oxidase. And so you have to take it with another chemical that's a monoamine oxidase inhibitor that allows the dimethyltryptamine to get into the body. So when the tea is brewed, it's usually brewed with 2 different plants. They don't always have to be the same thing, but one is the source of dimethyltryptamine, the other is the source of an antioxidant. If you take one by itself, it doesn't do anything. Harmine is a typical monoamine oxidase inhibitor. So as I said, used in religious rituals by indigenous peoples up here. There's a little bit of religious and quasi-religious use in the United States. I don't know how broadly it's used in this country. It's not really religious in the United States. It's people who want to try the cool drugs and are making up an excuse for the most part. at least with the people I've talked to. There's a couple small open label studies that have been done by groups in this country. I don't know this literature terribly well. So there's a couple small studies that suggest it might be beneficial, but they're not very big and there's no placebo control. So they're not rigorous medical studies in the way the depression studies I showed you, but there's a little bit, and that's promising and deserves more research. It is a little more complicated because they're two drugs. So anytime there are two drugs, it just gets a little more complicated to figure out the dosing and everything. So a little bit of ongoing work there. So I told you at the beginning, do I have the, yeah, here's the slide. So I told you that I was going to call these the classic or typical antipsychotics. We think that they act primarily by binding to the serotonin 2A receptor, and these are the atypicals. The atypicals act in a bunch of different ways. Okay, but this here, I'm going to talk, what am I talking about next? Peyote. Okay, peyote comes from a cactus in the United States and Mexico, northern Mexico, southern United States. And its active ingredient is this drug mescaline. Okay, and mescaline does act on serotonin, but not just at the 2A receptor. It also acts on another neurotransmitter in the brain, norepinephrine, which I'm not going to talk about a lot. This is the Peyote Cactus. This is used, I don't think this has been used for centuries. This is used as part of the Native American church, which is actually a church that was developed in the 19th century in North America in reaction to the extraordinary destruction of individuals and civilizations that was caused by European colonization. So people came together and so peyote use actually increased a great deal in the 19th century. So this isn't as ancient. But again, I don't know the anthropology terribly well. I'm sure it existed, it was used in previous centuries, but it became more prominent in the late 19th century. So this is where the peyote cactus grows, southern United States and northern Mexico, used by the Native American church, as I said. There are no controlled studies at all of peyote. And in fact, there's some work with mescaline, but not much as treatment. More people have looked at mescaline abuse. It's actually a problem to get peyote because the cactuses are all dying out because of over harvesting. So there's not, that I know of, there's a lot of anthropological interest in peyote and mescaline. There's not much clinical work that I know of. Maybe out there, if anyone knows, I'd love to hear about it. Other atypicals, very little clinical work with these others. There's a chemical called salvinorin A. It comes from chewing the leaves of a plant called salvia. Works in a totally different way. It binds to a receptor in the brain called the kappa opioid receptor. And it gives an experience that looks to me fairly unpleasant. Like, I don't fully understand why, but in low doses, some people use it recreationally. There's very few studies of therapeutic use. The kappa opioid receptor, this is a whole different story, but there's good reason to think the kappa opioid receptor has a role in major depression, and there are pharmaceutical companies that are looking at agonists, new drugs to bind to the kappa opioid receptors. There may be a totally different story there yet to be told. Ibogaine also has been traditionally used, but in this case by bark of a tree in Central Africa, and sometimes is chewed. It also binds to the kappa opioid receptor. There's a little bit of evidence in the treatment of addiction in the medical literature, but very small. Atropine has been around for millennia. If you put it in eye drops, it makes your eyes get bigger. It's actually used by ophthalmologists for that reason, but sometimes it's also used cosmetically by people who want their eyes to be big. And it's been used back in ancient Greece and ancient Egypt. Again, it has interesting effects on consciousness if used at higher doses. It can also make your heart stop and your blood pressure go down, so I don't recommend it. But it's in this universe of atypical psychedelics that people are using. Some other leaves and things that are traditionally used, but very little medical studies of these other substances. And I'm not going to spend any more time on them. What I am going to spend a little more time on are what's called the entactogens or empathogens. And those are drugs like MDMA, also called ecstasy. Okay, so this is a drug it's used recreationally. It's a party drug. It's been around for a long time. It does a number of things to both serotonin, dopamine, and norepinephrine systems. Doesn't have much to do with the 2A receptor, but it's famous. It's much less of a breakdown in yourself and a feeling of oneness and hallucinations than LSD and psilocybin, and much more about connection with other people. And that can include a feeling of friendship, of love. It can include sexual feelings, which is how it's sometimes used in the club scene. But it's called an empathogen because it builds feelings of connection, oneness, positive feeling towards people around you. And the big place that MDMA has been used, and there's a lot of literature on this, I'm not going to review it in as much detail as I did the psilocybin. I'm actually less familiar with it than I am with the psilocybin, but it's been used in the treatment of post-traumatic stress disorder, or PTSD. And this, I told you that Roland Griffiths at Hopkins was kind of a hero for going out and pushing against the field to get psilocybin research on the map. This group called the MAPS Cooperative, led by a guy named Rich Doblin, did the same thing for MDMA at about the same time, starting in the late 1990s. And Rich is kind of a kooky guy. He's used all the drugs himself, but he saw it help people. He saw it help veterans who'd been in wars, who had post-traumatic stress disorder, and he's really been on a lifelong crusade to put it on the map, and he's been successful. So it does a lot of things. It does bind to the serotonin 2A receptor, but it also blocks the reuptake, meaning the cells let serotonin out and then they suck it back up again. It blocks the sucking back up. So you get more serotonin. You also get more norepinephrine. You also get more dopamine. Does a lot of things. It's a dirty drug, but drug doesn't mean to be bad. Sometimes the combination of things is good. It's been studied for 2 decades by the MAPS Consortium. Now, in this case, I told you that in the studies of psilocybin with in alcoholism, they didn't just give them the drug. They gave them the drug in the context of a carefully structured psychotherapy that by itself might be beneficial. The question was, would the drug make it work better? That's what they've done here as well. They treat initially veterans and now other people with PTSD with MDMA in the context of careful psychotherapy. Now that's really good because people are getting individualized attention that's targeted to their symptoms, and it presumably is going to work better than the drug by itself. But now you think about, how do we get that out to the real world? It raises the bar that much more. Anyway, there's been a lot of studies of this over the years. There was a huge one published last year, and it's the only one I'm showing you. So the y-axis here is disability. Yeah, that works. So the y-axis is disability. Okay, it's how much are people struggling to do things in their daily lives. Down is good. And this is people who received MDMA plus psychotherapy compared to people who just received psychotherapy. Okay, clear difference. There's a little bit of a difference at the beginning, which complicates things a little bit. That's just, you flip a coin as to who gets in which group, and just by chance, the people were a little bit worse off in the control groups. That complicates things a little. But this is the PTSD symptoms. This is the comprehensive assessment for PTSD. Total severity, and they didn't have that problem here. These folks had similar severity of PTSD, and there's a pretty significantly improved difference. This is a 90-person study, quite large, and this got the attention of the regulators in the United States. They've got a second study going on just as big. If the second study looks just as good, this will get approved. It'll take a couple years, but this will get approved. So that's pretty exciting, and this has been a watershed in the study of PTSD. All right. So that is my tour, if you will, of the state-of-the-art of the use of psychedelic compounds of various sorts in the treatment of psychiatric disease. There's some other stuff I could talk about. There's some interesting work on psilocybin in the treatment of headaches, migraines and cluster headaches, which can be very severe disabling headache disorders. So that's it. There's some other, we're doing some of that at Yale. So there's some interesting other things. But I realize I'm already over. I was aiming for an hour so we'd have plenty of time for discussion. I'm already over that. So I want to wrap up with some what I consider the important open questions for the next 5 or 10 years in this field. Okay? So one is, I'm at heart a scientist. I'm a neurobiologist. I want to know how things work. And that's the first open question. How do these work? What do they do in the brain? How do they do what they appear to be doing? We know quite a bit about that at the receptor level, at least for psilocybin. I showed you some of the data for that. We're beginning to know a little bit about that at the neurobiological whole brain level, but there's more work to be done there. We don't know very much at the information processing psychological level. So that's an important ongoing study. Okay, this is a big one. What are and I've gestured to this at a couple of points during my talk. What are the relative contributions of pharmacological, contextual, and expectation to the experience? In the psychedelic literature, I meant to do that. It's all part of the plan. In the psychedelic literature, there's this concept of set and setting. Set is someone's expectations, what they think is going to happen. Setting is what's around them when they're having the psychedelic experience. So I told you there are some people who believe, or at least would like it to be true, that it doesn't matter. You give someone the drug. If I give you an antibiotic, as long as you take the antibiotic, I don't really care what your expectations are or what environment you're in. I just want to kill the bacteria, right? Most psychiatric conditions, that's probably not true. Like what you believe an antidepressant is going to do or whether you trust your therapist, that matters a lot. People don't talk about it as much as they should. People talk about it a lot for psychedelics. What are you expecting to happen? And what is the environment that you're in? If set and setting matter as much as some people think they do, then we've got a huge problem in terms of trying to get this out to the world. If they don't matter at all, then it's just like any other drug. And that's an important active question. I'm right in between. I think that people have to be in a psychologically safe space. They have to be able to relax. They have to be able to let a very disorienting experience happen and not fight it. And if they get in distress, they need someone there to just lay a hand on their shoulder, give them a drink of water. They need to be in a safe space. I don't think they need some complex, form of psychotherapy, nor do I. It's going to depend on the condition you're treating. But I don't think we can just sort of line people up in chairs and give them the drug. On the other hand, I'm not sure we need to spend 6 hours sitting with, 2 highly trained people who they've spent days getting to know. So I think I'm hopeful we can land somewhere in between. And as I've emphasized, that matters, right? This isn't a theoretical question. If these drugs work as well as the early studies suggest that they do, there's millions of people who could benefit from them. We don't have the machinery to roll that out with the way they're currently delivered. So this is not a theoretical question. So what can psychedelics teach us about normal human psychology, interpersonal relatedness, psychopathology, and even consciousness? This is something I haven't talked about that much, but it's something I'm fascinated by and hope to spend some of my time over the next few years getting to know better. I've started meeting with philosophers to talk about these questions, and I think these are really interesting. They're a little bit afield from the core question of how can we help people, which as a physician is a core question, but I think fascinating and important questions. And I've gestured to this a little bit when talking about the default mode. Here's a neurobiological observation that may be able to tell us something about the way we construct our sense of self. Maybe. So, interesting questions. What conditions can psychedelics treat? Who's most likely to respond and who's at risk? This last part's really important. So these are the conditions that I've told you about, that we've talked about over the course of the last hour. I'm going to kick that and make a loud noise if I'm not. So we're starting to have good evidence that we can help these people, especially depression and the despair of people near the end of that. That's where the data is the best. Here are the data. Oh, and PTSD, the data is quite good. These other ones are works in progress. But these are really carefully screened people. For the most part, they're not on any other medications. They have low or no suicidality. They have low or no tendency towards psychosis. Most studies, they even exclude people if they have first-degree relatives with psychosis, okay? They're not abusing any drugs, okay? Squeaky clean population. That's where you start. When you're trying to carefully study a new drug, that's where you start. That's not the real world. So something I'm worried about is when these things get unleashed upon the world with less regulation than they currently have. What happens if you've got a brother with schizophrenia and you take psilocybin, and you're depressed, and you take psilocybin? It might be wonderful. It might be a big problem. We don't know, okay? What happens if you have a little bit of cocaine or a little bit of alcohol in your system when you take psilocybin? We don't know, okay? So as we think about rolling these things out to try to help more people, not just the hundreds that have been in studies to date, but the millions who are suffering and we don't have good ways to help yet, these questions are going to become extremely important, and we don't know. And then finally, and I've talked about this a few times, how can we deploy these treatments to actually help people in the real world? And this is something that a lot of people, we're starting to do a little work we've got on this. We're trying to put together a training program to build a workforce, but it's a huge problem to actually get this out to help people if and when it's proven to the satisfaction of the community and the regulators. We've got a huge problem there. Okay, so that's it. Again, I'm happy to take questions and discussion. Apologize for cutting a little bit into our discussion period, but that's because I'm trying to talk slow. This is a list of some of my key collaborators. Ben Kelmendi, as I said, is the one who kind of pulled me into this area and has been a driver in a lot of our early work, and it's fun. So I'm grateful to him. Other folks in my research group have been involved, especially in the OCD work that I've shown you. Bunch of funders. My work is funded by a lot of people, government grants and various things. These are key individuals and organizations that have funded our psychedelic work specifically. And we're always very grateful to the patients who participate in our studies, without whom we can't do anything. We hope that it's always a two-way street, that they have a positive experience and ideally get some relief. But nevertheless, they're volunteers who are agreeing to do something that is unknown. And without them, the research doesn't move forward. Thank you all for your attention.
Thank you very much, Chris. Well, I have two questions before anybody raises his hand. One question is about, I was talking just last week to a guy that said that he used ayahuasca for 14 years. And he said that something that agrees with your comment there, that the setting of the experience is fundamental, that in his opinion, that it does not work if you say, sit there and take ayahuasca, that will not have the same effect as the practice. And here in Brazil, there is a cult, there's a religion that is quite strong, and even here in Belo Horizonte and so forth.
I've seen video recordings of those ceremonies. I've not witnessed one myself, but there's a lot going on there other than just the drug. The way I think about this, I do have colleagues, mostly older colleagues, who are really resistant to the idea that setting could matter, because it's a brain, it's a drug, and and I think this goes back in the history of psychiatry. About psychiatrists had lots of weird theories that were untested and unscientific and led the field in unhelpful ways for decades. And people in their 60s and 70s, people in psychiatry older than me had to fight throughout their careers to make psychiatry scientific. And I think that there's a fear that taking those things seriously will bring us back to the bad old days. I don't worry about that. And remember, I told you that my PhD work, my neuroscience work early on, was in synaptic plasticity. So if you give someone a drug that increases plasticity, it increases the ability of their brain to react to and encode information about the things around them. I'm not saying that's what the psychedelics do. This is a thought experiment. But if you give people a drug that does that, that's a perfectly neurobiologically respectable thing for a drug to do. But it's obviously going to make a big difference what environment they're in and what experiences happen, right? If you give them that drug and they get hit by a bus, they're more likely to have PTSD, right? And only hit a little bit by a bus. Whereas if you give them that drug and then they sit in a peaceful setting with people they love, listening to lovely music and looking at pictures of bunny rabbits, then that might be quite positive, right? So I have no problem with the idea that a drug that acts on a receptor in the brain and influences neurons is going to have systems-level effects and psychological effects that are profoundly influenced by the setting in which they're done. And but that's a strange idea for modern psychiatrists. And that's the balance that we need to try to find.
And the other question was about when you Lexapro, when you this type of drugs, do they work for the long time control of depression, panic, and those things, or not? If you take it for 10 years, does it make any sense?
The short answer is yes, often. It varies a little from case to case. In OCD tends to be chronic. It tends not, if it's bad, it tends not to go away on its own. So if I have someone who's had OCD for years, they come to me, I treat them with Lexapro, they get better. My advice to them is stay on it. And yes, it can be helpful forever. Sometimes people find that it helps for 10 years and then doesn't help, and it's a little hard to know. Is that because their brain adjusted, or is that because they just got older? It's hard to know, but it can. In depression, if you have someone who's never been depressed and then they develop a depression, either because of maybe a tragedy in their life or out of nowhere, but normally a depressive episode will last from 6 months to 2 years. You treat that person, they get better at 2 months, treat them for another year or two, and then you can stop. Because it's not a chronic condition. It was an episode, and that episode would have been over anyway. Some depression is chronic, but this is it varies. But yes, they can be helpful in the long term.
Questions?
1, 2, 3.
Três aqui na frente, né? Can you come here and identify yourself?
Okay, so my name is Arthur, and I wanted to ask that, I don't need this. So before you were saying that like there was like a cultural shift in the 70s that the psychedelics they started being associated with like politics, with like specific groups, and there was like this like attempt to like to exterminate like this research that it lasted like 50 years or so, and that now it came back. So what I wanted to ask is that since like the study of psychedelics is still like a bit like relative and not much is like absolute and like certified right now, like would there be a chance of like another like political and cultural shift that would maybe like start limiting this research again?
Yes, and that's something I worry about, and that's one I told you early on that while I personally have no problem with the recreational use of psychedelics, I'm not an advocate of getting it out of trying to get unregulated use widely. And part of the reason is because of as I said, I don't know who is safe and who's at risk. I don't know that yet. But part of the reason is because of the risk of another political backlash. I don't remember all the details, but one of the reasons psychedelics were so harshly regulated early on is because some rich, influential politician's daughter rolled off a roof while using LSD. I don't remember the details of this story, but this is something like that happened. It doesn't take that many people falling off a roof to completely change the conversation about the drugs. And so, yes, I do think there's a risk there. My feeling is that we've gotten enough momentum at this point, and there's enough, you can't question. I mean, even the best studies I've shown you are still fairly small. Like 90 people in a study isn't that big, but it looks pretty good. So I think there's enough more. I don't think we can assume everything is proven that I've suggested might be true, and I want people to be clear about that. But there's enough momentum that enough people think that there is something really valuable here. I don't think it can be shut down to the extent that it was in the 70s. I do think that there is a risk if things get out of control that there'll be a backlash. And so I think the community, the medical community, need to go methodically. And if we get in 20 years to it's available on the street corner and people are on their own recognizance to decide whether, I have no problem with that. But I think we need to go slowly and methodically.
Hello, my name is Bruna. Nice to meet you. Thank you for being here. Thank you. So there is a paper from a Brazilian scientist, Eduardo Scheinberg, and he discusses the framework of double-blinding psychedelic treatment clinical trials. Is this sensible? I forgot the word, sorry. Because it's another effect. So if we are looking for something that changed the way the person perceives itself, we need a new framework. Maybe. And there's another thing that I want to speak about, please. There is how much do you think that economical issues are holding back these progressive questions about health? I don't want to be the conspiracy lady, but I think I am.
How do you say conspiracy lady in Portuguese?
Because my question is about culture in the first place. Legal questions and culture. What is, in your perspective, the most important thing holding us back to have a new paradigm in health, mental health?
The second question is really big. I'm almost tempted to ignore it, but I'll try. But let me answer the first question, which is a really important one and smaller. So there are two problems with placebo control in psychedelics. I haven't read the article you're talking about, but I've read others on this topic. The first problem is the traditional clinical study. Nobody knows what drug you're getting. Some people are getting the drug, some people are getting the placebo. And the reason for that is so that everything else is exactly the same between the two groups. Both the things that we thought about and all the things we didn't think about. People's optimism or hope is the same between the two groups, right? People's how much time they spend with the doctors is the same between the two groups, right? That's why we do placebo-controlled trials. So one problem with placebo-controlled trials in psychedelics is, as I said earlier, placebo is kind of a joke. I mean, you take a pill and the walls start moving and you feel at one with your neighbors and you have, deeply meaningful memories of your childhood, and you take a different pill and, your heart rate goes up a little and you flush and then you feel fine in 20 minutes. These are not everyone knows what you're on. So that's a huge problem in these studies, even the most careful placebo-controlled studies that I'm doing. So that's a huge problem. One thing we do to try to deal with that is the people who are rating the symptoms, we have be blind. We do it over video link and they're in a different city. Right? So we can try to make the data as objective as possible. But still, the people know, the patients know. Another thing, a study that we're setting up to do, we don't have money for it yet, but we're setting up to do, is give people, so we know that ketamine can have an effect on depression, and we think that psilocybin can have an effect on depression. Those are kind of different. They don't seem to be exactly, they overlap in some ways. We're going to give people either ketamine or psilocybin and then switch. So everyone's getting something. Everyone's having an unusual psychological experience. We'll recruit people who've never had either one, so they don't know what they're saying. So there's ways to try to get around that, but it's a big problem. The other problem with the placebo control, and I think you were mentioning that you were referring to this in your question, is if the psychological experience is necessary to change, then so what does a placebo mean there? Like to fool someone by giving them the same psychological experience without the drug, if that were possible, it kind of doesn't make sense if the psychological experience is necessary to change. And that goes into, so there's some evidence in this depression work that the chances of depression improving correlate with the intensity of the psychological experience. There is evidence for that. That doesn't mean that the psychological experience is necessary. Right? It could be that the drug causes depression to get better and the drug causes a psychological experience, but there's no connection between the two. Or it could be that the drug causes a neural change, which necessarily is coupled with the psychological experience and is required. Or it could be that the drug causes the psychological experience, which is required. We don't know the answer to that. If it's this one, then yeah, that's a big problem for a placebo-controlled design. If it's this one, then it's not. You might be able to separate them. But so it's a very active problem, and it does require us to question fundamental assumptions about the whole idea in medicine of placebo-controlled trials. So I agree with that point, though I haven't read that article yet. I will look for it. What was the author's name again? Okay.
And others.
Sure. Okay. I want to come to another question and come back to your very big question, broader question. I think there was someone here.
Good afternoon. My name is Mateus. I am curious about what the studies tell about the risks in people with epilepsy or in people that are using antidepressants because of the serotonergic syndrome.
Yeah.
Is there any risk or something like that?
Yeah, so good questions. We don't know that much because people with those risks tend to be ruled out from these studies. So anyone with active epilepsy would be ruled out from almost every study. I don't know of a specific reason to be worried about giving it to someone with epilepsy, and I do know of some examples outside the medical literature of people who've used psilocybin and found it helpful who had epilepsy. So we don't have data. At least I don't know data. It may be out there. But it's not something I worry about that much. I worry a little bit more about blood pressure, people with heart disease, because it can cause blood pressure to go up. People with panic attacks, that can be a risk, but that's why we have people there to help. So there's some things I worry about. I don't worry too much about epilepsy, but we don't know. We don't have data. And that's one of the problems. As I said, the people who come into these studies are squeaky clean. They don't have any other problems. And so we don't know in the wider world. Your question about SSRIs, someone who's on Lexapro, for example, is an interesting one. We know that SSRIs, over time, in animals, cause a decrease in the serotonin 2A receptor. So we think that SSRIs might interfere with psilocybin. And that's why we require people to not be on meds in our study. And I told you that there were 2 of those people who didn't respond to my OCD study. One of them had just stopped her SSRIs like a week before. We think that her serotonin 2A receptors were reduced and she needed longer for them to recover. We don't know that. It's a theory. And there's animal work going on to try to address that. But you asked a question about risk and about serotonin syndrome. So serotonin syndrome is a rare but significant syndrome when people take multiple serotonergic meds and their brain gets flooded with serotonin, and it can lead to heart rate and problems, fever, seizures, heart rate dysrhythmias, blood pressure, bad problems. So that's a theoretical problem. We talk about it in our safety protocols. I'm actually not worried about it because psilocybin doesn't cause an increase in serotonin. It acts directly on the receptors, right? So it, if anything, competes with serotonin. So given what I know about what psilocybin does in the brain, I'm not too worried about that. It is something we talk about, we keep an eye out for. We monitor blood pressure very carefully. But I do know, so there was a recent review of 3,000 people who've received psilocybin in a carefully controlled medical setting over the last 20 years. There were no severe side effects. There was no serotonin syndrome. There was some elevated blood pressure, but nothing that became a big problem. In a really, really clean, carefully characterized population in a medical setting with pure drug of defined dosage with psychological help, it's very safe. In the real world, we got some work to do.
Hi, my name is Isaac. Thank you for being here. And I wanted to ask you, you said that patients have to be like squeaky clean, they have to be sober, they have to have low risk of psychosis. How does that tie into treatment for alcoholism? Because they...
Yeah, great question.
Yeah.
They, and honestly, I didn't do those studies, so I'm not completely sure. I think they had to not have alcohol on board at the time of dosing. Obviously, they were using in general, but I think they had to be sober at the time of dosing. But I'm honestly not completely sure. We're talking with the folks at NYU, so they did their little tiny study that I showed you data from, and then they did a bigger controlled study that I've seen data from, but I can't share them. And they're now talking about doing a big study at multiple different centers, and we're going to be one of those centers. So we're going to get more experience with that, but I don't know that right now. But it's a good question. And I'm not saying people have to be squeaky clean. I'm saying that's how people are doing the studies. And the reason, and I think that's reasonable. That's where you start, because the more heterogeneous, the more mixed the population that you're studying is, the more different sources of variation there are, the harder it is to find an effect. So starting with a uncomplicated population makes a lot of sense. Now, if you're treating alcoholism, that uncomplicated includes drinking, right? I think it's really important that we treat more complicated populations in a carefully controlled setting. I don't think it was a mistake to start with clean populations. I just think it's just the start.
Mais alguém? Vem aqui. I see that you are two very young people, and that you have a huge road to drive in this area. A lot of topics to exploit.
Hi, my name is Marcela. I'm studying psychology, and I wanted to know two things. The first one is, how can you prevent a bad trip in a person with depression when taking the drug? And the second one is, how can you measure the neuron degeneration that happens after? Because, in the common sense, we say that using drugs kills your neurons, and I wanted to know that. Thank you.
So the first question about the bad trip, it depends on what you mean by a bad trip. Many of the people who use psilocybin in our studies don't enjoy it. It isn't pleasurable necessarily. It's often very challenging. Often people have memories come up of bad things in their past that they have to struggle with. So it's not a euphoric sensation the way that some drugs are. So if bad trip means it's easy and enjoyable, well, it's often not. And I'm not sure it should be. Certainly, if we're talking about a psychological component to the causality, you might need to struggle with some hard stuff in order to break up pathological or maladaptive inflexibilities and come out the other end. So if that's what you mean by a bad trip, I'm not sure we should. If what you mean is feeling overwhelmed and terrified and not, even after the drug's out of your system, you still feel panicky, and that I think is all about controlling the setting. And so when I said, I think that I'm not convinced we need the two highly trained people that you have a trusting relationship, I do think you need to be in a psychologically safe space. I also think it matters, and so that if you start to feel overwhelmed, there's help there. I also think it matters how people are approaching the setting, the session. I suspect that if someone is being forced into the session against their will through peer pressure or a parent or a spouse, and they're terrified of what it might happen because of books they've read that made them, then probably they're more likely to have a bad outcome. And if someone is coming with trust in the people that they're treating and optimism that this will help them, then I think they're more likely to have a good outcome. So those are the two ways I think we can address bad trips in the second sense. I've forgotten what your second question was.
If you can control, if it can provoke any brain damage.
So well, was it brain damage, or do we know, is that your question, if that negative effects on the brain? Yeah. Well, so honestly, the way we're using it, I don't worry about that too much. But to test it, you can do cognitive tests, you can do brain scans, you can look in people who, and now that's hard to do in people you've treated clinically because you're treating them once or twice, right? But we follow them closely, but I wouldn't expect to see any damage. It's maybe in people who are abusing the psilocybin, or so there's work in ketamine. So we use ketamine as an anesthetic in surgery. It also is an antidepressant, okay? It can clearly be very useful and do lots of good things. And some people abuse it. And in people who abuse it chronically, it's fairly common in Asia. It's a drug of abuse, not so much in America. I don't know about this country. In chronic ketamine abusers, there is evidence of brain atrophy, of brain damage. So it's a difference between using it a couple times at a reasonable dose in a controlled setting versus chronic daily abuse. But it's a hard question. I mean, it's hard to prove a negative, right? It's hard to prove that if I give you a drug today and you feel better next week, that it won't cause your brain to shrivel in 30 years. I have no reason to think it will, but it's hard to prove, right? My feeling is that the way we're using the drugs is remarkably safe, safer than a lot of things that doctors do, and much safer than a lot of things that, you know, everyone in this room is probably doing over the next month. Safe, and there's very careful monitoring to make sure we don't develop evidence against that. Abuse of uncontrolled substances, I don't know. I would advise against it.
There's one there, and there's one from me. Chris, how do opium, cocaine, and marijuana relate to those psychedelic drugs there?
Yeah, it's an interesting question. Opium has no effect on the serotonin system. Opiates, fentanyl, has no effect on the serotonin system. They act by a quite different way. And I mentioned the kappa opioid receptor, but they act mostly on the mu opioid receptor. That's the one that makes people feel euphoric and has an anti-pain effect. So there's not much connection there. And the subjective experience that people go through is different. They're both obviously altering consciousness in some way, but it's qualitatively different, at least in the way people describe it. The cocaine has a little bit of effect on serotonin, but the most predominant effects are on dopamine and the ones that cause its addictive potential. So I wouldn't use cocaine and psychedelics at the same time. That gets a little messy, but I don't see a lot of overlap there. Marijuana is complicated because there's 300 chemicals in marijuana that do various things, and this batch of marijuana versus that batch of marijuana can contain different chemicals. The key chemicals, tetrahydrocannabinol and the other one, CBD, cannabidiol, work in a very different way, but they might work on some of the same neurons. I don't know of any evidence that they work in overlapping ways, but you could ask those questions in animals. You could ask those questions with brain scans. I do not know of any evidence that cannabinoids, that marijuana is good for depression It may be for PTSD, although the literature. In my country, at least, everyone's using marijuana. It's become legal in a lot of states. It's way out in front of the careful scientific literature. It's good for nausea. It's good for pain. It's good for appetite stimulation. It's good for muscle spasms, right? But it's being used for PTSD and OCD and all of these things that there's no evidence that it's good for, and it might hurt. So that's a big mess. But I don't know of any evidence that it works by the same mechanisms as the psychedelics.
Hello, I'm Paulo. I'm 17. I'm a high school senior year student. I'm kind of nervous, so I'm gonna read what
You're doing great.
Recently in Brazil, a famous journalist reported his positive experience with pharmacogenetic tests for depression treatment. I would like to ask you, in your experience, do you believe there is enough evidence to support this test as a tool to prescribe more assertive drugs for depression and other neuropsychiatry disorders?
Sure. It's a good question. The one place where I think it's useful, so mostly no. Okay, I think it has enormous potential. I think it ought to be able to help us. It makes a lot of sense that if two people have different forms of the serotonin 2A gene that lead to slightly different receptors, that might influence the effect of, that just makes sense, right? But the data isn't there. There's no data that we don't understand it well enough. The brain is complicated, the genetics are complicated, and drawing a connection we're not there yet. And when I have people come to me all the time with reports of their genetics and say, look, you should give me this drug, they're usually based on little tiny studies. Someone will publish a little tiny study in a 4th tier journal that no one else has read, and then the drug company will say, this proves that. Because they want to sell, not the drug company, the genetics company, because they want people to give them money to get their genetics testing, right? So going back to your question a little bit, but so mostly no. The one place where it can help, some of the genes that they test for are in the liver, and they regulate the enzymes in the liver that chew drugs up, right? So if you have a very active enzyme in the liver that chews up escitalopram, well, that means less escitalopram is going to get to your brain, and you might need a higher dose, right? That can be useful. Or if you have a very low, very inactive enzyme, I might give you a regular dose of escitalopram, but for you, it might actually effectively be a very high dose. You might be more likely to have side effects, okay? I don't do that for just general prescribing. What I do is I prescribe a small dose and see how they react. And if they have a side effect, I slow down. And if they don't, I go faster. And I find that more efficient. It's much cheaper, it's more efficient than the genetic test. So I think that there's enormous potential. I hope that in 20 years I can run a genetic test and that'll tell me got a 90% chance of responding to this drug and only a 10% of responding to this drug. So that'll help me with my decision making. We're not there yet.
And let's
Thank you for coming.
Move to the last question here. It comes from YouTube. A lot of the studies cited were non-controlled. The concerning issue here is the possibility of bias. Do you know of randomized control trials, blinded, being run about this topic, LSD, psycho?
Yeah, so that's a very fair question, and it is very common when we're looking at new drugs that someone has an observation. Someone writes on a blog, I took psilocybin and my OCD was gone forever. That catches my attention. And then that leads to an open label trial. And then that leads to a small controlled trial, which then leads to a very large control. And we're right in the middle of that in this field. Some of the data I showed, the 2 big trials in depression published in 2021, and the 2 big trials in late life existential despair published in 2016, and the MDMA trial in PTSD. All of those were careful, blinded, rigorous studies of a very high level of rigor. The other ones were not. The published alcohol study, not rigorous. The OCD work that I showed is controlled and sort of supposedly blinded, but it's small and not published yet. So those are not. But those 5 trials are good, careful, randomized trials. Now, this does go back to the issue of randomizing. Randomizing is easy. Blinding is hard. Mm-hmm. And a point that was behind the question that's really important is Well, another thing, we talked about how the people who are coming into these trials aren't necessarily representative of the broader population. They're squeaky clean is the word I use. They're also motivated. This is not a random cross-section of the population that's coming to try psilocybin. It's people who are excited about psilocybin, right? And that can absolutely influence results. Blinding, the randomization helps with that because both groups are equally excited about the possibility of psilocybin because it's random. That's why we randomize. That's why we don't choose. We randomize. So that is an issue. I think randomization helps with it. I think the blinding remains a big problem, as I talked about earlier. But it's a very fair point. I'm giving a positive story here. I came into this as a neurobiologist and a skeptic, and I have not tried any of these drugs myself. People sometimes ask me if I have not. I'm probably not going to anytime soon because I want to remain, careful and objective. I've been impressed by what I've seen. So I'm coming with an optimistic story, but it's really important. I've tried to do this in my talk, and I apologize if I have been a little bit too optimistic. We're in the middle. There are things that we're starting to think we know. There's a lot we don't. And we need to continue to go systematically and carefully. So I thank the question. That's my first question ever in my life from YouTube. And I thank the questioner for bringing up that important point to emphasize here at the end.
That was a good start. Oh.
I don't know, is he allowed?
The students should speak before because I was with him before. So my name is Henny. I'm School of Engineering, but coordinator of the graduate program in neuroscience here at university. So, Chris, I'm trying to think, but I couldn't find an answer. I don't know if you explained it or not during the talk, but you said that, so usually the drugs that we use in neurological disorders, they takes weeks to take effect. And you said that psilocybin, you can do it with one dose. How does it work in the brain? That's the first question I have. So how a single dose can make such an effect?
Yeah, so you're right that many treatments take a long time. Now, not all treatments. Treatment for allergy, can happen very quickly. So it just depends on the mechanism. Most psychiatric drugs take a very long time. You're absolutely right. Not all. Electroconvulsive therapy can work quickly. Ketamine as an antidepressant can work quickly. So we do have precedent. So we do have precedent for things working quickly. The way I think that works is I think that the drug makes a dramatic difference in the firing of brain cells and their connection with one another. And I've shown you imaging data for that. Then I think that leads to plasticity. I think that dramatic change, not necessarily up, some things down, but that dramatic change while the drug is on board in the organization of the firing of different parts of the brain relative to one another leads to plastic changes in the network. It leads to some synapses getting stronger and some getting weaker, which causes there to be continued change in the network after the drug is gone. So there's two phases to that. There's what does the drug do when it's actually there, at the level of network activity, these parts up, these parts down, these parts synchronized, these parts not. And then that leads to plasticity, to changes in the synapses which outlive the drug being on board.
And this can be a really fast process. I know that I did some research on that. We could measure it in half an hour, we could have something happen.
Yes. Yeah, so my guess is that if you gave someone psilocybin but blocked synaptic plasticity, then the effects wouldn't last. It would be an interesting experiment. It'd be a hard experiment to figure out exactly how to do that, but it's an interesting experiment.
So Stephen wants the microphone back, but I have two more. It's quick. Now there are quick answers. You showed some results with fMRI and with PET scan. I know well the functional resonate magnetic resonance image. I know it well. What's the difference between the results that you get with PET scan, the positron emission tomography, and fMRI?
Yeah, they ask different questions. The PET scan, you put a radioactively labeled tracer intravenously, and what you're measuring is where that tracer is sticking, where that tracer is concentrated in the brain and the body. And so the results and what you're measuring depending on what tracer you put in. So if you put in a tracer, if you put in radioactive oxygen or radioactive glucose, which tend to be distributed proportionally to the brain's activity, you get a signal that's quite similar to an fMRI signal. You get it has some time course differences, but you get a signal that's measuring basically the same thing as fMRI. But if you put in a tracer that sticks to serotonin 2A receptors, then what you're getting is a map of where the serotonin 2A receptors are. Has nothing to do with whether the neurons are firing or not. So then you get something fundamentally different from fMRI. So what you get with PET scan depends on the molecular nature of the tracer that you put in.
Last question, that's quick. fMRI is something that usually we can do it. It's harmless. You can do it through any ethics committee and things like that. How about PET scan? Can you go through ethics committee and let them inject some radioactive isotope in your blood without any problems?
There are well-established principles about how much radioactivity people can be exposed to in a year and over a lifetime, and what we do is way below. But you can't get 20 research PET scans in a year. You can get maybe 3, because what we use is about 1/10 of the dose that's considered the limit, which is about 1/10 of the dose that would actually be toxic. So we're using a couple % of the dangerous dose. But it is, people have to volunteer, and there's some theoretical risk of radiation exposure, but the amounts are quite low. The tracers are fairly high energy, but they decay. We use carbon-11. It decays in 20 minutes. So it's relatively high energy, but it's gone very quickly.
Thank you, Chris.
This is your job. I'm going to keep answering.
Thank you. You can talk later. Thank you. And then I would like to thank you, Chris. Let's thank Chris here.
Thank you all. It's been my pleasure.
E obrigado a todo mundo pela presença. O pessoal que está aqui, que está lá na outra sala, que não pôde, né? E lá no YouTube também. Muito obrigado. Tchau.
How was that?
It was great. Yeah. Very interesting.